In this video, we will discuss about DNA methylation work in cancer cells or cancerous cells. So, DNA methylation cause problems such as silencing of the gene, and in cancerous cells, this silencing might be related to uh, this silencing might be related to something that promotes the growth of cancer cells or prevent the cancer cells from being recognized by the immune system. So let us start with a condition where DNA is unmethylated and it is functioning normally. Consider this condition as the just after embryogenesis, and the methylation is observed on the CPG islands. CPG islands is considered as those sequence of base pair that has the abundance of CG sequence when read from 5 prime to 3 prime. And for more formal definition, we can consider it as in 200 base pairs sequence in DNA. If uh, presence of CG pair is more than 50%, then that uh, whole sequence of base pair is considered as CPG island. One thing we need to understood that when we are reading this CG from 5 prime to 3 prime sites. So let us come back to the topic. So the methylation of these CG sites or CPG sites is occurred via the enzyme that is DNMT3A or DNMT3B. So the red bulb shows the methylated site as this is CG sites. And because of this methylation, what happens is that the silencing of the gene occurs through. So this silencing of the gene is the final step. How it begins is that first the histidine deacetylation occurs, and when the histidine is free or is not acetylated, then the methylation of this histidine occurs and the HP1 binding occurs, and all these things will promote or in other words we can say prevent it will prevent the transcription of the DNA, as it will remove the transcription factor that are essential for the initiation of this transcription. So in other words, we can say that it will silence a part of DNA uh, where it is methylated or nearby its methylated sites. So this is how the DNA is methylated. But what happens when the DNA replication occurs? So in this case of DNA replications, the newly formed DNA is non-methylated. However, the already exist DNA is methylated. So in this case, what happens is that methylation is maintained with the help of enzyme this DNMT1. It will methylate it on the complementary side of the already exist DNA sequence. As we can see that it will methylate it in this side, this CG sites. Now it is read from 3 prime to 5 prime, the CG sites. And in this case, the methylation will continue in the cells even after the replication of DNA. And when this methylated will get removed, it will removed through epigenetic reprogramming, or we can say during embryogenesis. During embryogenesis, we will get a DNA sequence that is unmethylated. And however, in cancerous cells, this cycle will continue, and this DNA methylation will silence the gene that are uh, essential for working against the cancerous cells. And let us understand some in detail how this DNA methylation is prevented in normal cells. So this is the naked DNA sequence, and what happens is that in order to prevent the DNA methylation, there is a protein that binds to the DNA sequence, and it will prevent the DNA methylation. However, during aging, what happens is that the methylation begins at the border of this sequence, border of this uh, DNA methylated site, or we can say far away from the DNA methylated sites. And the reason for this is not yet known. And with time, what happens is that this CIMP sites, or we can say uh, the methylation will continue to increase further, and it will create methylation pressure, and it will create methylation pressure and remove these proteins that are preventing the methylation. The exact reason is not known, but uh, one of the reasons that is speculated is that mutation of these proteins might occur with time, and so they will not be able to prevent DNA from further methylation with aging. And what happens in the final state is that silencing of the gene occurs, as it is that is similar to what is happened in the left panel figure. So this is how the DNA methylation is achieved uh, when the, or we can say this DNA methylation is achieved with aging. It has been speculated, or it has been believed that this methylation begins at the border is mainly because of the replication, mainly because of the frequent replication of this DNA, and that leads to that leads to some mutation in this DNA with aging, and this will leads to this kind initiation of this methylation. As and this thing is similar to what happens in the cancerous cells. In the cancerous cells also, the initiation point is the single is the small mutation, and that will continue over a long period of time to develop tumor, and then metastasis of this tumor will leads to spread of cancer across different part of the body. So I hope you have understood how this methylation works, and its relation with the cancerous cells. If you need to understand this thing in details, you can go through the review paper, or the reference of that review paper is been provided in the description of this video. So that's all for this video. Thank you for your time.